Alright, so let's go over some of the basic elements of cell signaling. A lot of this you've already heard, and then we're going to talk about modes of cell signaling, the different types there are, and a few last concepts that we need to introduce. So, terms, hopefully that you've heard before. Ligand. Ligand is the extracellular messenger. Right? It's coming from either the bloodstream or from other cells. It's coming from the environment. It's basically signaling this cell what needs to happen. Right? And so we have various types of common ligands. We've seen neurotransmitters already, right? So that's sent from the end of this synaptic terminal from one neuron and signals the next neuron, and they can be excitatory or inhibitory. We have hormones, and these are going through the bloodstream, right? Similar to leptin that we talked about in video number 1. They can be secreted proteins from nearby cells. Even the extracellular matrix often can be a ligand. So if it binds to a receptor and initiates a signaling response, that extracellular matrix molecule is a signaling molecule. And then finally, direct cell-cell interactions. So you can get a signal from a cell where the ligand is not actually secreted, it's membrane-bound. And then cell-cell adhesion, when that membrane-bound ligand binds to the membrane-bound receptor on an adjacent cell, you get signaling. And this can actually lead to signaling both ways, because each of those two parts can be act like a ligand and a receptor, signaling both directions for the two cells. The receptor obviously is what's binding to and recognizing the ligand, and this is very tight binding often and very specific, so that it only recognizes the right signal. All right, so the ligand's response depends on the receptors. So you can have as much ligand as you want out here, but if the cell doesn't have a receptor to bind to that ligand, and therefore rally the signal and lead to a response, that ligand has no effect. So the ligand response absolutely depends on the presence of the receptor, and as we'll learn, maybe the type of receptor that's there. So the same ligand might actually activate multiple types of receptors, leading to different responses. Then, as we've talked about in the first video, since the receptor is stuck in the membrane, unless it's a steroid hormone, a nonpolar ligand, then we need a signaling cascade, right? This is the cascade of signaling molecules that relays the signal on towards the end. And this is going to lead to amplification in many cases. And then finally, it's going to activate the effector proteins, and these are the final proteins that actually activate the cellular response. So they can be the enzymes who have been turned on or off based on new phosphorylation or post-translational modifications. They can be the transcription factor that then turns on genes, etc. So the effectors are doing the final response. So what needs to happen for the ligand once it binds to the receptor for that signal to come inside? Well, we talked about this in the first video. That receptor needs to have a conformation change on the intracellular side based on binding of the signal on the extracellular side, so that the inside of the cell can realize, oh, there must be a signal bound. Right? So 6 modes of signaling. Basically, these are types of signaling. One is direct intercellular signaling. So this is a cell directly signaling the adjacent cell through gap junctions. That's the key here, gap junctions. Right? So can you think of any types of scenarios or types of cells where this might be important? Why do we need these gap junction signalings? Well, we need the gap junction signalings because it allows the intracellular environment of these cells to all be the same. So if one cell gets a signal, that's passed immediately to all the adjacent cells. So if this is a soluble ligand like calcium, for example, it's going to be passed immediately to all the adjacent cells, and then they all can work in coordination. And so some really common examples of this, one is the lung epithelia, right? So your lung epithelia is lined with cilia, so that if you swallow or breathe in something that you shouldn't, then that basically the cilia starts pushing it back out, and then you cough it out or whatever needs to happen. But the cilia needs to work when it's needed, first of all, then also coordinate in the same direction of movement. So if all of the cilia are just kind of waving randomly and not coordinated movement, nothing is going to move out. But if the cilia are moving together in a coordinated fashion, then it's going to pull or push that foreign object basically up and out of your airwaves. And so the lung epithelial cells constantly are communicating with each other, and these communication signals are going to basically control the movement of that cilia. So basically everything we've accidentally swallowed into our airwaves is going to come up and out. The more common one to think about is coordinated muscle movement, contraction. So for example, your heart muscles. It doesn't work very well if your heart muscles are beating at random. All right, that's AFib. And so if you have AFib, you're losing that coordinated muscle contraction that causes the atriums and the ventricles to actually push together, pushing the blood out. So these muscle contractions need to happen at the same time, and that's coordinated by the release of calcium through gap junctions that even are in the kind of pushed even further through these sarcoplasmic reticulum and the specialized aspects of muscle cells, which we won't get into detail here. But we talked about how calcium is used, or sorry, we will talk about how calcium is used to move muscles in Unit 5, right? But the coordination of it happens through gap junction intracellular signaling. So here we have some cardiomyocytes that are actually being grown in a dish, and those cardiomyocytes, they beat in coordination. So this is pretty low magnification, so each of these is an individual cell. And so you can see that they're all beating in coordination, even in culture, and it's because they create these gap junctions between all the cells. There's literally thousands of cells in this view, and that causes them all to beat in unified coordination, just like they would if they were controlling the beating of your heart. And this here is a pretty cool video where you have a kind of a set of cardiomyocytes, so this is a lot of cells together as well. And in these cells, they have injected a dye that basically reacts with calcium. And so when it reacts with calcium, you get chemiluminescence. And so you can tell if calcium has just entered the cells through chemiluminescence. And so they're going to create a stimulus up here, and you'll see that that calcium wave basically moves throughout all these cardiomyocytes fairly quickly because it's going through gap junctions and then basically just diffusing into each and every one of the cells. And that's what allows for that coordinated movement. So there's your stimulation, and then that calcium wave basically moves throughout all the cells. And it's moving that way because it is moving through the gap junctions. All right, the next type of cell signaling is contact-dependent signaling. So this is when, as I was talking about, the receptor is membrane-bound as normal, but the ligand is also membrane-bound. So in order for this signaling to happen, these two cells have to come in direct contact with each other. All right, so see if you can think of any examples of this type of signaling that occurs in the human body. Well, there's actually quite a few of them, but the examples that I will give are based on the cell growth. Right? So contact inhibition. So tumor cells should, actually no cells should grow when they are too tightly packed. Right? And so when they're too tightly packed, those two cells are going to contact each other. Both, in this case, the ligand and receptor are both signaling, so the signaling is bidirectional, and both cells will be told not to divide because it's already too tightly packed. And then finally, the immune system, T cell activation occurs this way as well. So let's go to just a little bit of the idea of T cell activation. So obviously we have our DNA, and our DNA is being transcribed to mRNA, and then that gets made into protein. But if the body wants to know what types of proteins are being made inside this cell, if the T cell is on the outside, it needs to basically get a smattering of the proteins that the cell is making. Right? And so the cell will actually take a very small percentage of the proteins that it makes, chop them up, and load them onto the cell surface on MHC receptors. Right? So basically, each cell is presenting a representative portion of all the proteins that it's making inside the cell. And that's what the T cell comes along, and through contact signaling, right? So with the MHC, the T cell receptor, and that whatever ligand, whatever kind of bit of protein peptide is being presented at the cell surface, this will create signaling. And if this is deemed to be self, non-foreign, the T cell will move on. But let's say your cell is infected with a virus, a la the COVID virus, hopefully not. But if you have coronavirus in here, that coronavirus is hijacking your cell to use your cell to make proteins out of its genome, right? And so in that case, hopefully this cell will randomly take some of the proteins of the virus and chop it up and just present that at the cell surface. And now the T cell recognizes this as foreign, especially if this T cell has adapted and been cloned to really recognize coronavirus. And so in this case, now that cell will know that this is foreign, and it will signal this cell to undergo apoptosis. So this is contact-dependent signaling. The T cell and the cell it's perusing needs to be in direct contact for that signaling to occur. So if the cell is infected, T cell would be activated, signaling in this direction towards the T cell, and then the T cell, once activated, will signal in this direction to kill infected cells. So that's a little bit overly simplistic. All right, so those of you that have taken immunology probably know this in much more detail than this, but that's the general idea, and that's an example, a good example of contact-dependent signaling. All right? Then we have autocrine signaling. All right, so what does autocrine signaling mean? You've probably heard this before. What does that mean? Well, in autocrine signaling, we have the scenario where the ligand is being made by a cell, transcribed, translated. It's being packaged into the ER, so it has an ER signaling sequence, then sent on to the Golgi and then secreted. So this is exocytosis of this ligand, right? And so autocrine releases the ligand, and it's going to bind to receptors on adjacent cells, and even to receptors on itself, and that will cause signaling. So autocrine signaling releases a ligand that signals adjacent cells and signals itself. Okay. So again, we can have inhibition of growth. Autocrine signals are going to reduce growth in nearby cells and also itself. A few other examples there, but again, autocrine is a signal that affects nearby cells and the cell that released the signal in the first place. Then we have paracrine signaling. All right, so there's a very subtle difference between autocrine and paracrine signaling. What can you spot here? Well, the ligand secretion is the same. The fact that it binds to a receptor and that's what causes signaling is the same pretty much for everything. But the difference here is that the cell secreting the ligand doesn't have the receptor. So it's not signaling itself, it's only signaling cells around it that have the receptor. Right? Autocrine signaling itself and others, paracrine only signaling the others. So some examples of this. So blood vessel growth. If you have hypoxic tissue, whether that's a tumor that's hijacked your system and trying to get blood vessels in it, or just tissues that's growing and so forth, well, that tissue needs to signal the endothelial cells, the blood vessels, to grow into that tissue to prevent hypoxia. Well, there's no point for that ligand to signal the cell itself because the cell itself is not part of the blood vessel system. So it's only going to signal the blood vessels, and those endothelial cells in the blood vessels are going to have the receptors, but the cell signaling it, the tissue signaling it, will not. That's paracrine signaling. All right? So it does not affect the cell from which the signal is secreted. So what factors do you think limit autocrine and paracrine signaling to local? Well, this is actually one of the concepts we'll talk about. So the ligand's half-life will limit it. So if the ligand degrades rather quickly, it's not going to be able to diffuse very far. The extracellular matrix in the region can actually limit it. So this is basically limiting that diffusibility of that ligand if the extracellular matrix is very dense, or if the extracellular matrix has proteins that kind of bind and competitively inhibit the ligand from moving further, that can inhibit it. And also just the amount of cells with receptors. If there's a lot of cells that are going to be reacting to the signal nearby, less of that ligand is going to make it further. So those are probably the three biggest things, although there's more. The half-life of the ligand, the extracellular matrix, and what the extracellular matrix has in it, and then kind of the number of receptors and so forth. But this will also determine how wide-ranging that signal is, how far it can diffuse, and how far cells away from the signaling cell will still respond to it. All right, then we have synaptic signaling. In a sense, this really is paracrine signaling. You have one cell exocytosing, releasing a ligand that binds to an adjacent cell, not itself, and causes signaling. But because this is so important and kind of unique in terms of the type of paracrine signaling with the neurotransmitters and action potentials and so forth, it's given its own classification. Right? So This is just where neurotransmitters are released. They're going to bind to those ligand-gated channels that we've heard about before in Unit 2. And then if this is enough to hit threshold, then the voltage-gated channels will activate, and that'll send a signal down the next neuron. Or the neurotransmitters might signal the muscle to move, or whatever its target cell is meant to do, if it's kind of the last neuron in that cascade. And then we have hormonal signaling. Right? This is called endocrine signaling, endocrine meaning hormonal signaling. So these are signals that are sent through the bloodstream. Right? So which is faster, neuronal or endocrine signaling? And why? Well, your neuronal signaling is much faster for the reasons we learned in Unit 2. Those neurotransmitters are ready to be secreted immediately. They hit the ligands that are very close in the adjacent cells, and then that's going to lead to a very rapid action potential if it hits the threshold and so forth. This is extremely fast signaling. We know that because our reflexes are very fast, and that's a good thing. If we sat there, leaned on a hot stove, and it took us about a minute for our signals to basically reach the brain and then back down and us to realize that our hand has been burning up, that's problematic. But we know that as soon as you hit that hot stove, it's a reflex pulling it off. So, on the other hand, the hormones have to travel through the blood system, and then they have to signal the cells. And we're going to talk a lot in the first synchronous session about why some cells respond to the hormone and others don't. Because once this hormone's in the bloodstream goes everywhere. It now has access to every cell all around the body. So why do some cells respond, others do not? And why do some respond at different levels of that hormone? But this is going to be a slower signaling, so that hormone has to travel, hit a receptor, and activate cells in that regard. Much slower process than neuronal signaling. All right. Then, even when that effect has happened on the cell, those effects can be slow or fast. Right? So we've got transcriptional regulation and post-translational changes. So which one of these is that very rapid effect in a cell? Well, we've talked about this before. Post-translational modifications are there because they are very rapid responses to change in environment. And the change in environment in this case is the ligand binding the receptor, and that can activate a signaling cascade and then basically post-translationally modify a protein so that it's pretty much immediately on or off or whatever change was needed. This is almost an instantaneous change, and it can last for a little while, but it often is reset, as we'll find out in this chapter as well. The slower one is transcription. So that signaling cascade is going to activate a transcription factor, which now is going to enter the nucleus. It's going to transcribe, leading to the mRNA, and then you have protein synthesis. Now you have a new protein you didn't have before. But we're talking in terms of milliseconds here, we're talking minutes along the way. And this usually lasts longer as well. But both of these lead to altered cell behavior based on the signal and the events needed for the cell to adapt to. Cells must react to many signals at once. We're going to do this in this unit, and you see this all the time. We often think of a single ligand binding to a receptor and a single signaling cascade and therefore an effect. And that we do that so that we can really understand some of these signaling cascade events, right? But the reality is that there are literally thousands of signaling events going on at once. So thousands of receptors, thousands of ligands, and multitudes of signaling cascade molecules. And this gets extremely complex because the signal coming in through one receptor might cross-react with a signal coming through another receptor, and yet the cell does so very well at getting the right response based on these things. And so I want to make sure that this is not taken lightly. This is not a trivial thing. The cell is responding to literally thousands of signals at once. The signaling cascade molecules are working all simultaneously, and you're getting the correct result. And oftentimes, it's the multitude of signals that really determine what has to happen. It's the combination they're in. So this is just an overly simplified kind of version from your book, but this is what it's talking about. If you have, for example, signals A, B, and C, that might culminate in survival signals telling that cell it should survive. And this often is the case too, because if you are in if the cell is growing in the wrong place or in a scenario where it shouldn't live, then one of these will be gone, and that cell will die by default, right? But here we get our combination of survival signals. But if you add 2 more signals to that, now suddenly these extra 2 signals not only kind of cause this to survive, but now growth and division, right? Replacing dead cells, for example. But the same 3, ABC, with 2 different ones, in this case F and G, that can cause differentiation. It can cause changes in gene expression and allow the cell to mature and differentiate into a different type of cell. And then we talked about it, the lack of these survival signals may cause apoptosis by default. Right? So it's the combination of the signals that are really going to determine what this cell is doing. But the cell has to understand all the signals at once. And that's difficult because the cell can't understand, it doesn't have a brain. This is all just chemical reactions coming together and causing the right cellular response or the right combination of cellular responses. Right? And we've seen this before. So if we have ligand A, it's going to bind to its receptor and lead to a signaling cascade. If we have ligand B, it's going to bind to its receptor and lead to a signaling cascade. And that might ultimately combine to lead to protein Y being active here. Because A might activate this phosphate group, B might activate this phosphate group, and if this is a protein that needs both phosphate groups on it to be active, then this is only going to respond when you have A and B. Maybe something different responds when you only have A. Something different responds when you only have B. Those 2 things may not respond when you have both, but Y does. And this is what we talked about from the very beginning in Unit 1, when we talked about how this is like a binary switchboard. So a single protein can have multiple inputs where all of them need to be satisfied in order for it to be active. All right? And here's the Sark kinase, for example. This phosphate had to be removed, so this phosphate's inhibitory because this phosphate binds kind of this domain and keeps it from being free to interact with a ligand. So when this phosphate's removed, this actually is now free and it can bind to another protein, and that protein quaternary structure is required to activate it. So maybe another signal caused the transcription and activation of that second ligand that has to bind to this domain. And then maybe there's a third signal yet that has to add this phosphate so that the active site is open. So all 3 signals need to occur, all 3 ligands need to have been there, all 3 conditions need to be met for this to be active. There's your binary switch mechanism. All right, so let me know if you have any questions on that, because that's pretty important. This is, in very simplified manner, kind of how these signals can combine together to activate the right signaling molecules and therefore the right cellular response. It's right back to that multiple inputs. You need some phosphate groups added, some phosphate group not added. You need protein binding, you might need GTP bound, you might need a multitude of things, and those come from a multitude of signals. And we call that integrating all the signals into the right response. All right, we're going to talk a lot about all of these concepts, get some pretty intricate detail on these things, but this is the general ideas of what we're going to talk about in Unit 4. Thank you.